Okay, so in lecture number 8 of this module, I would like to spend more time to talk about one specific issue in single-cell RNA-seq, which is the sparsity of the data. Okay, so in the kind of grand scheme of the single-cell RNA-seq workflow, the computational question regarding the sort of improving the quality of the data kind of resides right here, okay, in this step or this component. So once we have the gene cell count matrix summarized from all these large collection of reads, then we already know that this data matrix has very high sparsity. So in the literature, there actually have been a series of papers developing computational algorithms to impute these zeros so that hopefully the quality of the data will become better and the subsequent analysis will become easier. That's sort of the hope of these imputation algorithms. So but before talking about any existing imputation algorithms, I'd like to quickly kind of introduce one definition for dropout or sparsity, and also talk about why they occur and how big of a problem they are, right? So dropout is, according to actually one of the reference papers that I forgot which one. So according to that paper and a couple of others, dropout is defined as a, dropout refers to a phenomenon that occurs in single-cell RNA-seq where a gene is observed at a moderate level in one cell, but this gene is not detected in another cell of the same cell type. So, for example, if we think about, say, MS4A1, which is a B-cell specific gene or B-cell marker gene, it is supposed to be expressed in all the B cells. But if we look at all the B cells in the data set, and for this particular gene, we'll see that many of them express this gene at a pretty moderate level, but then some of the B cells, they show zero expression or zero count for this particular gene. And this is, I think in many people's opinion, this is a technical artifact, right? Oh, and one question is, how do we understand the word moderate? So there actually are gene markers, cell type specific genes that are really, really highly expressed. For example, there is a gene that's called FCGR something, which is a monocyte marker. And this gene is pretty much detected in 99% of all the monocyte cells. So, so definitely for really highly expressed genes that are cell type specific, those genes, it's possible that those genes are detected in majority of the cells of that cell type. But for cell type specific marker genes that do not show very high expression, then sometimes we miss it in the cells belonging to the corresponding cell type. So of course, moderate is a kind of hand-waving word. I don't really have a good way to define. For example, count 5 is moderate or count 10 is moderate. I don't really have a good way to concretely define that word. Okay, so now in terms of why dropout occur, okay, so first of all, a single-cell RNA-seq is measuring mRNA content or signal at single-cell level, and the amount of material there is very small for us to catch, and hence, any like data acquisition technology will have its detection limits, right? So low amount of mRNA content and detection limits. This is one reason why dropout may occur. And another reason is there may be insufficient RNA capture. So for example, when we have within an oil droplet a bead and a cell, right? And when we lyse the cell, then the mRNA contents are available for the bead to catch, but is the bead able to catch all the mRNA molecules there? Well, the answer is no. I think I've heard numbers like 10%, 20%. So that's, I guess, roughly the capture efficiency of single-cell RNA-seq. That means, well, there are a good amount of mRNA molecules, that are actually there but not captured. Right? And a third reason is stochasticity of gene expression. So and this refers to the fact that if a cell needs to express a protein at a very low level, okay, then the cell actually does not need to maintain an active mRNA copy for that corresponding protein or that corresponding gene at all time, because mRNA's molecules, they degrade faster than proteins, right? So the cell can generate an mRNA molecule, and then say some protein molecules are made, and then the mRNA is degraded, but the proteins are still there, doing what the cell needs them to do. So the cell does not need to produce more of it, and hence, the cell doesn't really need to keep producing mRNA molecule for that gene. But maybe after a while, these proteins are all gone or all degraded, and the cell feel the need to generate more. Then this gene will be expressed, and the mRNA molecule will be present, right? So in that case, although the cell actually needs this gene's corresponding protein, right, to be there, to work like all the time, but then the cell does not need to maintain an mRNA molecule at all time. And really, it depends on when we measure the cell. Right? If we're lucky, we see some counts for this gene, but if we're not lucky, then we don't see any. Okay, so that's another reason why we see a lot of zeros, or why we may expect a non-zero value to be seen, but what we see in the data is 0. So in terms of how big of a problem it is, it's actually really hard to say. Well, if we look at droplet-based techniques, for example, 10x genomics, a typical data set will contain, I would say, more than 90% of zeros. And if we look at fax-based techniques, for example, SmartSeq2, then the percentage of zeros or the sparsity of the data is lower, okay, is around 80%. But then, are all these zeros kind of dropouts, or are all these zeros problematic? Are some of these zeros, really supposed to be zeros? It's something that's very difficult to really determine, and hence I would say, if we say how big of a problem is sparsity in single-cell RNA-seq, then I can give you these numbers, 90% and 80%. But then, how big of a problem is dropout, right? Is how many zeros there are that's caused by these sort of technically undesirable artifacts. I really don't have a good way to quantify that. Okay, and in terms of how are people handling dropouts, or how are people handling the sparsity in the data, so we've seen in the SIREP pipeline or the clustering pipeline, we can select highly variable genes that are, in my opinion, less affected by dropouts, and we can perform dimension reduction, for example, PCA, t-SNE, UMAP, and these dimension reduction techniques, they are, in some sense, borrowing strands and averaging across different genes. In a way that follows the correlation structure among the genes. And these two steps can help us to reduce the sparsity. Of course, we can actually try to develop imputation and apply imputation algorithms to directly try to change and update the data in a way that zeros are replaced by appropriate values. So in this slide, when I talk about dropout, sparsity, and zeros, you kind of probably already hear me not being super consistent about what these different sort of phrases mean, right? There actually is a fairly recent review paper from U of Chicago, and one big message in that paper is that dropout is a bad word to use, because in the literature, the word dropout has actually been used to refer to many different meanings. So in some papers, dropout basically is equivalent to all the zeros we see. Okay, so all the zeros, the sparsity is called dropout. But in some papers, dropout refers to the kind of undesirable zeros. So the in this data matrix, there are so many zeros. Some of the zeros probably should be non-zeros, right? But they are zeros because of technical artifacts. So those undesirable zeros is referred to as dropouts. Okay, so that's another definition that people use. And the third definition of dropouts that people use is that dropout is they say dropout is a phenomenon that actually affects all the values we see, the zeros we see and also the non-zeros we see, because of insufficient RNA capture. So for example, in our data matrix, we see a non-zero, say gene A in cell number 5, the count is 10. That's what we see from the data. But because of insufficient RNA capture, the number of mRNA molecules for gene A in cell number 5, right? Maybe it's more than 10 molecules. We just didn't catch them all, right? So so dropout may be affecting actually all the numbers we see, even the non-zeros. And if we want to do imputation, maybe we should make adjustments not only to the zeros, but also to the non-zeros we see. So given these kind of 3 different definitions of dropouts out there, this review paper strongly recommended against using this word. And that's why, now when I sort of try to present this work, I often try to use the word sparsity to refer to sort of the zeros and the problem here, so that hopefully it's less ambiguous. All right, so in terms of how people are handling the sparsity of the data, as I mentioned earlier, is actually more always about sort of borrowing strands across similar stuff, either similar genes or similar cells. So one approach is, again, selection of highly variable genes, and then PCA, dimension reduction, followed by subsequent analysis. And because this PCA is sort of each principal component is averaging multiple cells that are relatively correlated with each other. So then this kind of averaging is able to reduce the sparsity. And then after we have the PCA space, we can follow up with a UMAP kind of visualization or community finding kind of clustering analysis. And another approach is imputation. And as I mentioned earlier in the previous lecture, imputation is about borrowing strands. So for example, if there are multiple genes correlated with each other, then we can use the average of these genes to inform how we want to impute each of these correlated genes. And same goes for the cell axes. If we have multiple cells belonging to the same cell type or even the same cell state, then they probably should show very similar expression profile. And if many of them express one particular gene, but then some of them don't, then maybe those zeros, right, should be filled with non-zero numbers. And then after imputation is performed, hopefully we have a better version of the data that serve better for subsequent analysis. Okay, so the existing imputation methods, there are many out there, and today what I want to do is I want to pick 2 of them for this presentation. One is MAGIC and the other is called SC-impute. Okay, so the reason why I choose these 2 algorithms as representatives is because, well, first of all, they are the two relatively early algorithms that got popular, and second, they actually represent 2 distinct kind of ideas about how dropouts should be handled or how sparsity should be handled. So in the MAGIC algorithm, the idea is that dropout is something that affects all the values we see, the zeros and non-zeros. And therefore, what that algorithm does is that it tries to impute the entire data matrix. So the zeros are imputed into non-zero values, and the non-zero values are also elevated to be larger values. Okay, and in the work called SC-impute, the basic idea is that, well, when we look at all those zeros, only a subset of them are caused by technical issues, while many of those zeros are probably supposed to be zeros. Therefore, when we do dropout, we should first identify which zeros are supposed to be zeros, which zeros are what we want to correct, and only correct the latter kind of zeros. Okay. All right, so my opinion, I'll put a question mark here, and I'll just say, for now, my opinion is somewhere in between, but we'll circle back to this after we talk about these 2 algorithms, MAGIC and SC-impute. Okay, so MAGIC is I don't know why this is happening, but looks like my PowerPoint crashed again. Give me one moment. All right, so MAGIC is an algorithm developed by a group based in Colombia, and it was published in Cell, so it's very high impact journal. The analysis pipeline is listed here. So the starting point is, again, this gene cell count matrix, followed by library size, normalization, log transformation. And then after getting this transformed and normalized version of the data, the authors decided to perform PCA and only retain 70% of the variance. So at the end of the day, they they tried multiple data sets, and the number of PCs kept in each of those data sets ranged between 10 and 20. And once they have the data reduced in the PCA space, now very similar to the DPT approach, the authors computed pairwise Euclidean distance between each pair of cells, and then computed this affinity matrix. Okay, so in MAGIC, the formula for the affinity matrix is slightly different from DPT, but is essentially doing the same thing. Okay, so once the affinity matrix is obtained, then what they did was they further processed this affinity matrix by zeroing out very small affinities. Okay, so remember that in this affinity matrix, for two cells, I and J, here X and Y are just cell indices. Okay, so for two cells X and Y, or two cells I and J, if they are very far away from each other, then their affinity, because of this exponentiation, their affinity is very small, very close to zero. And then in this paper, what the author did was they just went one step further and say those very close to zero values, let's just replace them by zeros. Okay, so this is effectively done by constructing a k-nearest neighbor graph using the affinity, so that very low affinity pairs of, cell pairs with very low affinity, they're not connected, and hence their connection is zeroed out. Okay, and then they get this A matrix. This matrix now is, well, actually, again, pretty much the same as the UMAP's k-nearest neighbor graph, weighted k-nearest neighbor graph, right? Remember what UMAP does is that it constructs k-nearest neighbor graph and then use this kind of formulation to generate weights. Okay, so that's this A matrix. Now, so far, actually, we've seen that multiple algorithms like ISOMAP, t-SNE, UMAP, MAGIC, and diffusion single time, all of them, they're kind of using very similar ideas about how they consider distances, long distances and short distances, and this kind of exponentiation. Okay, so here they have this matrix they call A, which is an adjacency matrix of a weighted k-nearest neighbor graph constructed using this kind of affinity formula, very similar to UMAP. Okay, and then they compute A plus A transpose to make sure that this matrix is symmetric, and then they turn this matrix into Markovian probability transition matrix M. So the M matrix here is essentially the T tilde matrix in the DPT algorithm. So in the DPT algorithm, what they did was they computed T tilde plus T tilde square plus T tilde cube all the way to T tilde raised to infinity power. Okay, and in this paper, okay, so what they did was that they have their T tilde matrix here, and they only raise T tilde matrix to a certain power T. Okay, they only consider T step transition, not a cumulative effect of many different possible steps. Okay, and they do have a algorithm that determines what is the appropriate value of T. Okay, and this sort of T tilde raised to certain power, this matrix would represent what they call the appropriate steps of diffusion. Okay, and their basic idea is that once they have this graph, right? This matrix, this k-nearest neighbor graph that connect cells that are similar to each other. Then in order to impute one cell, we can look at this cell's direct neighbors, just its k-nearest neighbor. But we can also look at this cell's k-nearest neighbors plus those neighbors' neighbors. So this cell's two-step neighbors. We can also look at maybe this cell's three-step neighbors or t-step neighbors. And if we determine an appropriate t, then we'll say that for this particular cell, right, although its gene expression profile is very sparse, but I can look at this cell's t-step neighbors and use all those neighbors to help this cell to impute its gene expression profile. Okay, so at the end of the day, this m raised to the t's power, this matrix will basically describe this t-step transition for each cell, how much help it should get from all the neighboring t-step neighboring cells. Okay, and then we just apply, or we just multiply the original sparse data by this mt matrix, right? So that for each imputed cell in the new data is a weighted linear combination of itself plus its t-step neighbors. And that's how imputation is done. And now because the raw data, okay, the imputed data is derived by the raw data multiplied by a matrix. So potentially all elements in the data matrix is updated, okay? And therefore, this approach, when it tries to update and impute the data, it will mess with both zeros and non-zero values. Okay. All right. So that's how magic works. And then in terms of SE impute, this is an algorithm developed by a statistics group in UCLA. And here is kind of their general pipeline. So starting from the raw data, the basic normalization and log transformation is the same. And then this is the paper that I mentioned earlier. So they also did PCA, but then they said they only wanted to keep a certain number of PCs to retain 40% of the variance. Okay, and then, well, when they get the data into PC space, they also do the pairwise distances, and then they remove some outliers so that they are considering kind of fewer cells. And once they have this pairwise distance matrix, so this paper did not do that affinity thing. And what they did was they just took this distance matrix and then applied the spectrograph partitioning algorithm we discussed to obtain K cell clusters. So this number of clusters is something that they specify. Okay, this is something that we need to specify if we want to use this algorithm. So how this process works is that, well, starting from all the cells, we can apply spectrograph clustering once to get two clusters, and these two clusters, one is bigger than the other. Okay, then what they chose to do is they will just say, let's take the bigger cluster, apply spectrograph partitioning again to divide the bigger cluster into two pieces. Now we have three clusters, and then among the three, whichever is the biggest, they apply spectral analysis again to break that into two pieces. Now we have four clusters, and we can keep going, right? Until we have K clusters defined. So this is kind of their approach of defining clusters. But I would say that, this part of the pipeline, we actually don't have to follow their approach. If we just follow the standard Sarette clustering pipeline, it's actually, I think, also okay. The key point is that what they want to do is they want to divide their cells. They want to cluster the cells into, well, clusters where each cluster contains cells that are hopefully of the same cell type, or at least highly similar to each other. Okay. And then once they have the clustering information, they come back and they take the raw data, or actually the normalized data, and then they organize cells according to clusters. Okay. And then what they want to say is that, if these, let's say these first group of cells, if these cells, they belong to the same cluster, then they should share a very similar gene expression profile. And then we can start to say, focus on just one gene and look at how these numbers, okay, the observed counts for one gene across cells in the same cluster, we can look at how these genes distribute, okay? So what they say is, if, not how these genes, sorry, how these values distribute. So for one gene across cells in the same cluster, if we look at the expression value and we see a distribution looking like this, okay? So that means across all these values, okay, we either see very large, relatively large values of expression, or we see zero, okay? Then in this case, it is likely that all these zeros correspond to technical artifacts, because for this gene in this cell cluster, okay, whenever we see this gene expressed, the expression is really high, right? And whenever and that means in cases where we do not see this gene being expressed, we see a zero, though that zero may be technical artifacts. Okay, but if the distribution of these counts, okay, look like this, okay, so that means for this gene across cells within this cluster, sometimes we see zero, sometimes we see non-zero, but when we see non-zero, the value we see also vary a lot. So that means seeing a zero is also possible that, well, zero is kind of within the variation of the non-zero values we see, and therefore we don't have strong evidence that these zeros are not supposed to be zeros. So they have these mathematical formulas, or these statistical formulas to compute, well, using the intuition I just mentioned, they use these formulas to compute what is the likelihood of a zero being a sort of technical artifact, okay? And that's this Dij here. And then later what they do is they will say only for zeros whose corresponding probability of being an artifact is sufficiently high, only for those zeros, they're going to do imputation and replace those zeros by non-zero values. Okay? And the way they do this is by some kind of linear regression analysis, which is, well, quite tedious to explain, but it's just done by sort of looking at cells and genes where, well, for a particular gene, it's looking at cells where this gene is expressed, and then see how that expression relate to other genes, and then use that relationship to make predictions on cells that show zero expression for this gene. Okay? So essentially, it's just a linear regression model for learning, and the same linear regression model for predicting what those zero values should be. Okay, but the key point here is that zeros are, I mean, not all zeros are imputed, and under SC impute framework, the non-zeros are not touched at all. Okay. Okay. So one comparison, well, because SC impute and MAGIC are the first two algorithms on imputation, both of them are highly cited, and a lot of subsequent imputation algorithms compared with these two. So one observation, and kind of my biased observation and interpretation of these two algorithms, is that if we are looking at a data set that's more trajectory-like, then MAGIC is probably a better algorithm to perform imputation. But if we are looking at a data set that's very much trajectory-like, containing distinct cell types, then MAGIC often would induce kind of spurious correlations, and S-impute is a better algorithm, in my opinion. Okay. And also, I also mentioned this earlier, there has been a benchmarking paper comparing existing imputation algorithms. And it's a paper coming out of a Johns Hopkins group. And their conclusion was that imputation is good in terms of its ability to recover correct values of recover kind of single cell expression values that would align better with bulk RNA-seq data, and also better recover correlation among genes. While at the same time, majority of these imputation algorithms actually do not improve performance of downstream analysis, because downstream analysis, they often already have some components of dealing of how to deal with sparsity in the data. Okay. Now, circling back to this slide I mentioned earlier. So the two general ideas of thoughts about dropouts and how we should deal with them. We either try to impute the entire data, the zeros and non-zeros, we elevate them all, or we only try to touch a subset of the zeros that we feel are more likely to be artifacts, right? So in my opinion, I think the first approach of imputing the entire count matrix, I think is too much. And I do believe that some of the zeros we see in the gene cell count matrix are supposed to be zeros, and we should not touch them. And my personal biased opinion, I think a fairly large percentage of the zeros we see in the data are probably supposed to be zeros. Okay. And while at the same time, the second approach of imputing and updating only a subset of zeros may not be enough, because many of those non-zero values there, due to insufficient capturing, they probably should correspond to large numbers if the library preparation, the whole process, has better efficiency. And therefore, combining these two thoughts, I think a better approach for imputation should be that we try to elevate all the non-zero values, and we also, and for the zero values, we only impute a subset of the zeros that are supposed to be artifacts. Okay, so however, I think a big problem to sort of really materialize this idea, right, down here is that, for example, the process of SImpute, how it identifies which zeros are likely to be artifacts. It needs to rely on the non-zero values to identify, possible artifacts of zeros. But then if we say that, hey, we also don't really want to 100% trust the non-zero values we see, then we kind of also, in some sense, lose our basis to identify which subset of zeros are problematic. So I actually have sort of this slide from 2 years ago's SISG presentation, and the same question here. So I've been actually thinking about, if I want to implement and design an imputation algorithm that behave like this, how would I do it? And 2 years has passed. There's still a question mark in my mind. But anyway, I think this is something interesting to think about, and I have not given up on it yet. All right, so far, what I have presented, right, is kind of various strategies to address dropouts or address the sparsity. And well, because of the high sparsity in single-cell RNA-seq data, it is actually pretty natural to treat this sparsity as a challenge of the analysis, and hence a problem to be fixed. However, later when I was thinking about this, one idea that came to me, since everybody viewed this as a problem to be fixed, is it possible that I can kind of go the other way around and try to explore an opposite view and utilize the sparsity of the data as a useful signal? Okay, so this is a wild idea, but later I followed it up, and then I actually indeed develop something that's useful, which is what I'm going to show you in the next slide. Okay, but before going there, I actually want to sort of take a slight detour and tell you, one kind of general formula for computational innovation that I feel very useful. This is not something that I came up with. It's something that a mentor told me when I was a postdoc fellow. So what that mentor told me was that, if you want to do something that's truly innovative and transformative in a specific topic field, then one possible idea is to go and read top seminal papers on that topic, and then try to identify the common assumptions that everyone make, and then try to challenge that assumption. Can you try to come up with a story without that common assumption that everyone needs? If yes, then it is likely that you're going to come up with something that's fundamentally different from what everyone is doing. So to me, I think this is probably a general kind of mindset that everyone is thinking about in their sort of subconsciously, right? But for me, when that mentor spelled that out to me, it was super helpful. And in my own research career, I've been, trying to, very often I've been trying to sort of think about that general formula and how that can apply to the specific research topic that I'm working on. And this is actually one example, right? So when I notice that everyone is thinking about dropout as a problem, then can I present a story that use dropout or sparsity as a useful signal? All right, so then what I did was that I say, given a single cell RNA-seq data, let me just binarize it. Okay, so I turn all the non-zeros into just ones, and the zeros are still zeros. So this particular binarization step actually removes majority of the quantitative information in the data, and what's remaining is just where the zeros are, where the non-zeros are. And then I had a series of steps of building gene-gene relationships, and then community finding clustering, which we discussed, and then build cell-cell graphs, do the same thing to derive cell clusters. And it turns out that just based on the binarized version of the data, it is possible to identify cell clusters that correspond to real cell types, which is something that's super interesting. So here is a diagram that represents my work in this reference paper. Okay, so starting from the raw data, we first binarize the data so that we only have zeros and ones in here. And then the first step is I computed a gene-gene similarity matrix. So here, this box, okay, the dimension of it is the number of genes by number of genes. And each element describes the co-occurrence of two genes. By co-occurrence, I mean a statistical quantification of how likely two genes are detected in the same set of cells, okay? And also kind of undetected in the same set of cells. If that's the case, then these two genes would co-occur and the corresponding value here will be close to 1. But if two genes, they are mutually exclusive from each other, so they're detected in distinct set of cells, or their detection are just random. Sometimes they're seen in the same cells, sometimes they are only one of them is seen, right? So in that case, the two genes do not co-occur and their co-occurrence value will be close to 0, okay? So, and this matrix, we can think of it as an adjacency matrix of a gene-gene graph, where each node is a gene, and then each value here is an edge, the weight on an edge connecting two genes. And after, we can use this to define a gene-gene graph, and then apply community finding algorithm to identify gene clusters, okay? And for each gene cluster, here I use a different word, I call it gene pathways. And then for each gene, well, let's just call them gene pathways. Okay? So for each gene pathway will contain genes that tend to co-occur with each other. They tend to be co-detected in the same set of cells. And then I will use these gene pathways together with the binarized version of the data to do dimension reduction, where each gene pathway is one dimension. And here the numbers here, okay, represent the activity or the detection activity of this pathway in each cell. So for example, if a gene pathway contains 100 genes, then in cell number 1, 90 of these 100 genes are detected according to the binary version of the data. Then we'll put a 0.9 here because this gene pathway of 100 genes, its activity level detected is 90%. Okay, and if in another cell only one of these 100 genes are detected, then we have a 1% in here. Okay, so for every cell, I can quantify how many percent of this pathway is detected, and that gives me sort of this one row of values here. And for each of the P gene pathways, I will do the same, so that the dimensionality of the data now becomes P dimensional, and along this axis is still just the number of cells. Now, once the dimensionality of the data is greatly reduced, then I decided to compute the Euclidean distance between each pair of cells, just 2 columns in here, okay, to form this distance matrix. And once this distance matrix is formed, I applied community finding algorithm again to define cell clusters, okay? And let's say it gives me K cell clusters, okay? And then for each of these K cell clusters, what I can do is that, for example, cluster number 1 contains 500 cells. I can actually take these 500 cells and go all the way back and say, if we start with the raw data of only these 500 cells, so cells in just one cluster, right? And then go through the same process and see whether I can identify gene pathways and also further cell subclusters, okay? And I can just keep iterating. Now, stopping criterion is that, for example, if I'm looking at a set of cells that are really coherent with each other and do not contain subclustering structures, then there will not be any gene pathways identified, because all the genes are just always detected, for example, or always undetected. Then there's no correlation among genes at all, and hence no gene pathway can be identified. And we know that I know that I should stop. Okay, so here is kind of the same graph, but with some data overlaid to it. So this is a PBMC data set with almost 3,000 cells. After binarization, the dimensionality is still the same. And then co-occurrence matrix is gene by gene. So it's 32,000 by 32,000. And then after applying community finding, what I noticed was community finding algorithm actually identified many gene clusters. Okay, and those gene clusters, majority of them are very, very small, containing only like 1 gene or 2 genes or 3 genes, right? So I apply the threshold and I say that for small, tiny communities with less than 10 genes, I'm just gonna ignore all of them. So then it turns out that for this data set, I only saw 4 gene pathways, and each of them contain, well, 500 genes, 200 genes, and this is less than 100 genes, maybe 80 genes and 200 genes, right? So now I have these 4 pathways or 4 gene clusters. And if I reorganize my raw binary data, okay, so that genes corresponding to the same cluster come together, I also actually reorganize the cells according to the final clusters I saw. But here from the binarized data of these selected genes, we can already see pretty clear kind of block kind of structure, indicating that there may exist distinct cell clusters, right? So once we summarize these selected genes into pathway level information, then this is even clearer that there exists one group of genes whose activity for the first pathway is really high, but whose detected activity for the rest of the pathway, and then for the rest of the cells, right? So for these cells, the first 500 cells, their detected activity for pathway number 1 is very high. These cells have high activity for pathway number 2, and these cells here, they have high activity for pathway number 4, right? So when I take these data to generate cell-cell similarity matrix, and then community finding easily identified 4 cell types or 4 cell clusters. And then after that, each of these 4 cell clusters, by the way, the last one is very small, so it's very difficult to see its pattern, but just trust me, the pattern is very, very clear. So after that, I took cells in each of these 4 clusters through the same process again. Some of these 4 clusters are further divided, some of them are not. So here is kind of the full results. So the first analysis gave me 1, 2, 3, 4 clusters. Cluster 1 in the next iteration was further divided into 2 clusters. And then this cluster in the next iteration was further divided into, again, 2 clusters. And I did perform further iterations on these clusters, but then was unable to identify any further pattern to suggest further division. Okay, so so this process, this is the co-occurrence iterative clustering process based on the binarized version of the data only. Okay, and then co-occurrence gave me 1, 2, 3, 4, 5, 6, 7, 8, 9. It gave me 9 clusters for this data set. Now I also applied standard SISG pipeline, which is based on the quantitative data of highly variable genes, right? Remember what SISG does is take highly variable genes, PCA, and then community finding. So, and the SISG clusters are listed on this axis. So here, this is sort of the confusion matrix in between the two algorithms, and we see a lot of agreements. Although there are some disagreements, but one thing we can conclude here is, in general, SISG-based clustering based on quantitative information of highly variable genes versus co-occurrence-based clustering based on just zero, non-zero sparsity pattern of all the genes. These two approaches, they actually generate highly similar clusters. That means in terms of defining cell types and cell clusters, the sparsity pattern can be almost equally informative compared to the quantitative data of highly variable genes. And this is actually something that's super surprising. Okay, so I don't want to spend time to explain these other plots, but just to say that the gene pathways or gene clusters identified by co-occurrence process, they are indeed highly relevant to the underlying biology. And some of the clusters identified by co-occurrence, because we are using binary pattern of all the genes, right? So those clusters actually cannot be distinguished in SISG because it only focuses on highly variable genes. Okay, so I guess overall the message I want to deliver with this analysis in this paper was that the sparsity pattern, the zero non-zero pattern that everybody view as a problem, it actually can be a useful signal for defining cell types. So here is another analysis based on another data set, okay, where when we compare co-occurrence clusters versus kind of non-SISG clusters, we see this staircase pattern again. And when we compare co-occurrence clusters with the developmental time corresponding to all these cells, we again see this nice clear staircase pattern showing that clusters defined based on these binary data is meaningful. All right. So, and here is sort of one last result of co-occurrence clustering performed on the tabular murus data. So this is a very large data set obtained by multiple mouse organs and is profiled by two different techniques. One is 10x genomics and the other is SmartSeq2. Okay, so now co-occurrence was applied to these two data sets separately, and we see just, let's say, some nice results. Okay, and one interesting observation is that when I look at the detected pathway activity in between the two data sets, so this here in each plot is a organ, okay, profiled in tabular murus, and the horizontal and vertical axes corresponds to some derived co-occurrence-based features. Well, actually, the average pathway activities derived from the two data sets. So when we see, these dots following the 45-degree line very closely, that means the pathway activity-based quantification of these two data sets gave us pretty much the same signal. And there's very little batch effect in between the two data sets if we look at the binary sparsity pattern. But if we look at the quantitative aspect of the data, then we'll see that, comparing to the two data sets, the 10x data set versus the SmartSeq2 data set, the variation is a lot larger. Okay, so that means, and this shows that the gene pathways identified by co-occurrence analysis and also the pathway activity level is actually highly conserved in between the two data sets. Although there exists actually good amount of batch effect in between the two data sets, they are generated by two different technologies. Even the percentage of dropout is, or percentage of sparsity is very different. But the pathway activity level of quantification is somewhat invariant to the batch effect, whereas the quantitative information of the highly variable genes is highly influenced by the batch effect. Okay, so this is yes, this is my last slide. So the take-home messages here is that, well, there exists a lot of imputation algorithms. I would say if we just want to do imputation, okay, then and then try to identify maybe a better gene expression profile, right, and identify better gene-gene correlation, then MAGIC and SIMPUTE and other imputation algorithms probably are going to be useful. And I would say MAGIC is more useful when dealing with trajectory-like data, and SIMPUTE is more useful when dealing with cluster-like data. While at the same time, if our purpose for imputation is to improve subsequent analysis, then imputation may not be able to help a whole lot. Maybe just use kind of existing pipelines based on the raw data before imputation is likely equally good. Now, I've also shown that opposite from the common view of the literature, right, the sparsity of single cell data can actually be a useful signal that we can use to identify cell types. And this is sort of shown in a paper I published 2 years ago and the results that I showed in the previous slides. And in fact, there were two other papers published around the same time that also hinted on the usefulness of the sparsity of the data. So there was, one genome biology paper that used the dropout as part of the features they want to use to do clustering. And there was a commentary saying that single cell RNA-seq may not be that zero inflated. And the zeros we see there, maybe many of them are meaningful zeros. So collectively, these 3 papers have been cited more than 300 times. So I think the message of potential utility of the sparsity is kind of already out there. But so far, I haven't really seen many kind of follow-up papers to further explore this idea. Okay, all right. So this is the last slide, and I'll stop here and see whether there are questions. All right. So I'll just go from the bottom. From Emil, there's a question about, if we look at the co-occurrence pathways, okay? So these gene clusters that tend to co-occur with each other, can we use this as information to differentiate meaningful dropouts versus unmeaningful dropouts? I think the answer is definitely yes. So and this idea for identifying and if you follow such an idea to identify, say, which zeros are likely artifacts, right, then your idea actually will be, I think, very different from what's implemented in SIMPUTE. So actually, I would encourage you to kind of follow your train of thought and try to see whether use kind of co-occurrence pattern of genes can help you to differentiate zeros that are supposed to be zeros versus zeros that are artifacts. I think that this actually is a very nice idea. Right, and then moving up from Xiulan, her question is whether or not co-occurrence is available in R. Unfortunately, it is not. It is only available in MATLAB. But if you go onto this reference paper, you will see a implementation over there and also examples you can run. And now moving up, a question from Justice. So he's a little bit confused and wanting to ask, what do I seek to achieve with co-occurrence clusters? So actually, it's very good that you remind, this question reminds me of a comment. So thinking about the co-occurrence clustering pipeline, right? The first thing that was done was binarization of the raw data, and this step actually lose a lot of information. Okay, so certainly I'm not trying to say that everyone should cluster cells using co-occurrence. That's not what I'm trying to say. In fact, this step is, I think, should be considered a very poor pre-processing step because it loses information, right? So with this whole paper, what I wanted to show was, even with such a bad choice, pre-processing choice, so that I lose a lot of information and I only have the sparsity pattern available to me. Even though there is such bad pre-processing, with an appropriate algorithm, we can still recover useful biology. And hence, whatever information left in here can be viewed as useful information. Okay, so this first step is sort of a strategic choice to kind of put myself in a corner and then ask whether I can still do anything useful, right? So at the end of the day, I think this whole paper is trying to just deliver the message that the binary sparsity pattern in the data can be a useful signal, right? And therefore, what everybody is doing right now, right? When they do clustering, they use quantitative information of just highly variable genes, use that information to do clustering. But how about the binary sparsity pattern in the other genes? Those actually can also carry useful information, and therefore maybe a better algorithm, or at least an alternative design of clustering algorithm, could be an algorithm that take quantitative information of highly variable genes and then also binary sparsity pattern of maybe the rest of the genes or all the genes, and then combine these two pieces of information in some meaningful way to define clusters. Maybe that will be somewhat helpful. Okay, the co-occurrence clustering algorithm itself, I actually don't think people should just go and use it. Okay. All right. So let me stop the recording.